So mimic3 only is an option, but limits the number of awesome genomic data sets that we can bring to bear. So if you're not interested in this more broadly, however, connect with Na to look at, in my group, to look at ROSMAP cohort where we have.
I'm here.
Oh, hey. And phenotype and single cell RNA-seq data. So basically now we were just talking about sort of phenotypes and EHRs. So, you know, this is one possibility where, you know, a RES could get access to some of that data. All right, study transfer learning with convolutional neural networks and computational biology problems. Try using existing models from other domain problems and some novel genetic problems.
Manol, before we transfer to idea two, can I just ask you a quick question?
Of course, of course.
So while I understand the issue of data, and that might limit this project, as a research question overall, is the combination of EHRs and genomic data of interest?
Humongous.
Okay.
Humongous. Yeah. Yeah, absolutely. So, and I think this project would be pretty awesome. So basically, the challenge is that we don't have hundreds of thousands of people. We have, you know, 600 people.
Yeah.
Interactions between phenotype, genotype, RNA, etc. Yeah. So I completely agree. So I should say the questions. Okay. So transfer learning, scan through the conventional kernel sizes. Yeah, love it. You know, challenge, make it more specific, find right data, but general principle is great.
Yeah, one of my ideas was to find some classic problem in computational biology that involves convolution, and then just to go in different pre-trained models and from different domains, pictures, graphs, or whatnot, and see which ones have the best accuracy on genomic data. I don't know if that's a list or not.
The challenge is that pre-trained models will probably, you know, from other domains will probably not apply. However, so you could do transfer learning on a large data set of healthy folks where you train TFDS models, transcription factor binding site prediction models, and then use pre-trained model on a disease cohort. Right? So basically, you know, this could also apply to the EHR space, right? Where you could pre-train on mimic, e.g. pre-train on mimic, apply to, you know, ROSMAP.
Yeah. Okay. There's no like possible different problem space that would have similar functions that the network is trying to map, similar to EHR that you'd know better than me.
I doubt it, but I mean, in the EHR space, sure, potentially, but in the genomic space, it's not like you're going to take something from images and then apply it there. So another application could be image analysis in ROSMAP and integration with genomic data. I think that is actually the killer project.
Okay.
Yeah.
So Manolis, how about this can apply to the integration between single cell attack and single cell RNA?
Oh, yeah. I know that the imaging is something that probably appeals to RES much more because it's sort of more phenotypic. And then this is too genomically. So basically, I feel that sort of integration between two different genomic data sets probably appeals to him less then integration with something that's sort of quite orthogonal, right, Arez?
In general, but I'd love to discuss that more, Nassan, and
Yeah.
see if it might, draw me in.
Yeah, good. I love it. Okay, simple computational rules that might govern DNA mutational processes, something like very small genomes, these ideas regarding a rule-based system to try and find the existence of such rules in DNA mutational processes. So, C project from, oh gosh, a gazillion years ago. Isao Fujiwara, gosh. Okay, so I think that's a bit too theoretical. I think we already have some, awesome suggestions there. Ari, did we do yours already?
Thank you very much, Professor.
Of course, thank you.
No, we didn't.
Okay, so multi-decade-long attempt to domesticate foxes. With the past few years, the author published a paper to commit QTL of the difference between tame, aggressive, and domesticated foxes. We analyzed the data set of QTL analysis, but not GWAS. Sometimes the two methods identify different regions. Only included a relatively small number of haplotypes. And we need QTL and limit their selection to loss identify the pre-obstructed behavior genomically. Cool. So my guess is that they were grossly underpowered.
Sure. Yeah, I think they were. I didn't think about that at the time.
From the corner of my eye, I thought I saw like something flying around, but I'm guessing it's the reflection of a bird from the outside. Weird. Of GWAS loci from previous studies. So yeah, my guess is that a paper that makes it thus far probably has explored all avenues and sort of they arrived there by necessity, not by choice. So, it's kind of cool. So basically, perhaps one way you could do that would be to look at GWAS overlap with other comorbid traits, e.g., schizophrenia, bipolar, neuroticism, etc., from UK Biobank, and search for common heritability between the
That's a good idea. So just do a QTL, but with different loci also.
Yeah, to epigenomic data sets to see what tissues and cell types are implicated, etc. Does that make sense?
Yeah, yeah.
Oh, great. Aggregate the Chimera Net single data sets as a guide and train the CNN on that. The paper featuring considering chromatin stability to improve the model performance. Sounds great. Authors of the paper feature here, not to mention the chromatin stability data. We can evaluate the magnitude of the effect. Yeah, I like it. A bit of a risk because it might not work. But certainly worth trying. And then use approach for several problems to boost power and see which problem it works well in. You see what I mean? Basically, I'm saying, yes, sure, try it for this, but try it also for a few other places where you can sort of use feature information to improve power. And if this one in particular doesn't work, then yeah, you could use other ones. But so basically, cool computational trick can be reused for other applications where it might work. Sounds good?
Yep.
One or more of the ASCA activity of regression analysis for some potential specification. Oh, I like that. I like that. So basically, it's an unbiased search for that. So basically, search rather than starting from chromatin. One or two cats. Systems use different molecular mechanisms for cutting. It seems very plausible that there are common factors influencing guide activity. Yeah, sure. Yeah. So, you know, a bit too technical, I would say. So I think idea number one is, you know, probably the best. Sounds good, Ari?
Yeah, cool. Thank you.
Okay, Kathleen, did we go over yours?
We haven't.
Cancer cells have more variability in the TADs compared to normal cells, and the chromatin structures surrounding certain loci are altered in cancer cells. Is it possible to use this information to classify cancer as normal cells? Cool, I like it. Potential methods include generate a graph where nodes represent high CV data. So the problem that I see, but challenge, is you're classifying a set, not individual ones. Let me explain what I mean. So, you know, if you basically have a set of cells and you say, you know, I have 20 cells here and 20 cells there, what is their variability? The one with the higher variability is probably cancer.
Yeah.
But if you take a single cell, you don't know its variability compared to other cells. Do you see what I mean? So basically what you're describing is a set classification where I already give you a bag of cells and you're like, are they all cancer? And a bag of other cells, are they all, you know, normal? Do you see what I'm getting at?
Yeah.
So,
So that was actually my concern as well, is that I won't be able to like isolate the structure that I'm looking for to differentiate them.
Yeah. Though you could look for cross comparisons of each cell of interest to be classified versus all other cells, both tumor and normal. So that's sort of one option. But in general, more generally, heteroscedastic models can be broadly applicable through different application domains. Okay, so potential methods include generate a graph where nodes represent high C read data, cancer and normal, focusing on certain chromosomes, and edges refer to similarity between nodes. Yeah, sure. Incorporate the chromatin structure. So see, generate a graph with nodes and yeah. Okay, does that help?
Yeah.
Okay, so idea number two, extend the study of cancer classification of single cell expression data by using a graph attention network. Oh, I love it. Cancer. Yeah, so that's a fantastic idea. I love it. Efforts to profile multi-omics data in single cell, either using available data for further analysis to gain more insights or implement different integration methods of information. I don't know one, certainly. So what do you mean by latent information here?
So things like we always, whenever we choose method, we always talk about, like in MF, we want to look for the latent components to describe the data. It's kind of like reducing the dimension is kind of like a way to emphasize that hidden representation.
Yeah.
But honestly, for this one, I'm not. It's like it's actually still really general, because like the paper that I'm looking at, it has like some data sets of like integration of single cell data, and each of them have like different functionalities. So like, I'm still like honestly not really sure about this one.
So these are just some ideas about latent information. So, you could use Go terms that are associated with a particular phenotype, or a latent space of gene-gene correlations in general, or perhaps some modules of genes agnostic of phenotype. Does that make sense?
Okay.
Awesome. Let's see, Maya.
Thank you.
I'm just looking at people on my screen and going through your ideas. I hope that's okay. All right. In the genus Peromyscus, monogamy has evolved at least twice. Cute. Independently. Although behavioral traits may differ between monogamous and promiscuous species, some physical traits vary as well. I have several folks from Harvard. Are you one of the folks from the?
Yes, I'm
yes.
from the Hoekstra lab.
Say it again.
The Hoekstra lab. Yes.
Yeah, exactly, from the Hoekstra lab. Exactly. Yeah. Like, wait a minute. This is how come you even know about this? Okay, good. So I apologize for having to look it up, but yeah. So.
Oh, that's fine.
Behavioral traits may vary between monogamous and promiscuous species. Some physical traits vary as well. Tested size, increased spur production, spur aggregation. All these tests are from two promiscuous species and two monogamous species. And look for signals for selection. I love it. Totally love it. Determine which genes may be experiencing selection. Yeah, I totally love it. Yeah, so basically, yeah, so you have all the right data sets. It seems like it's the right thing, and you have phenotype, and then you could also integrate with GWAS studies from their orthologous genes in human and find out what other traits these may be involved in. Right?
Thank you.
I love it. All right. Now I'm going to have to Google this one. Oh my God. Okay. Soft lies in family. That's her husband. Sister species, Leconte and Pinectum larvae. Melanin-based spotting patterns that exhibit variability within both species. Previous work showed that the genes associated with pigmentation are likely common. Known genes involving pigmentation to soft lab, but the protein coding regions might not explain specific variability. Cis-regulatory genes have been shown to explain much pigmentation variation. As a guide, look for potential regulatory regions. Oh, I love it. So yeah, so you can use, oops, what did I just do? You're suggesting and commenting. That's not what I wanted. Yeah, so you obviously know about modern code.
I've never heard of that.
Okay, so in a previous life, I actually, sort of built a circuitry of the Drosophila genome. So basically, you can look for, yeah, so, and then did you look at the 12 flies compared to genomics paper?
No.
Okay. Right?
Yes, thank you.
Good. Neodyprion. There's a lot of interspecies variation pigmentation using known pigmentation-related genes along the SCBI data. Compare color genes across the species and look for signature of selection, protein coding difference that may cause functional differences. Yeah, sure. Answer, but a bit less opportunity for data. Okay, does that help?
Yes, thank you.
Awesome. So let me look at the list of folks here. So Farhan, did we go over yours already?
No, we haven't.
Okay. I can't find your name on there.
I couldn't find my name too. I don't know what's.
Oh no. Oh no. So oops. My guess is that there was a deletion, like an evolutionary deletion. Something was deleted here. So Tessa, can I ask you to look at the history and then search for Kodai and then look for history for what was there and was deleted?
Yes, I can do that.
Okay, why don't you start doing that? It seems that we have Farhan's full thing, but,
Yeah, but
check whether there was something missing at the end of Reina's.
Sounds good.
Okay, perfect.
can we briefly talk about it, Manuel? Because I talked with Suvi and LY about the project of isomers,
Yeah,
microRNA.
Yeah.
And we're working on some of the data that we have, and I think it's really interesting. I just don't know how to form a team, and like if anyone is interested in working on those kind of projects for understanding heart failure. Would it be possible for me to work by myself, or because I'm already like?
I mean, I would strongly urge you to make an effort to find teammates. And
Mhm.
maybe, you know, Tessa, what we could do is have a project pitch kind of, you know, session where basically people pitch their projects and folks sign up for them. So, I mean, you know, what kind of sucks is that everybody has these awesome, totally awesome projects. So everybody will want to work on their own. Maybe it was a bad idea to sort of have people write their top 3 choices because chances are that one of them is awesome to start with. So, but my guess, again, Tessa, sorry for picking on you, it's just that I don't see the other TAs, is that you probably have some folks who haven't submitted their top 3 ideas, and maybe I can start being a little more critical. But you know, I think it's a great suggestion, and you know, I can't force anybody to work with you, but I think we can certainly advertise projects that folks can sign up for. So we haven't made too much of an effort beyond just these team building breakout sessions for creating this. But what I would do, Farhan, is search through the profiles of people and just search for the word heart or search for the word disease.
Unfortunately, I couldn't find anything. People are more interested in cancer and neuroscience. No one cares about the heartless people.
Yeah, it's not like it kills a lot of people, right?
Another thing, oh, sorry. On Piazza, I don't know if you've seen, but there's also something where you can just like post like for teammates, like looking for teammates, looking for a group.
Yeah.
I think that's something that Brian put together, so that might be something of interest to try and find some people who might be interested in your ideas.
Yeah, I agree.
Sounds good.
So look at those resources and then let's see what you find.
Sure. Thank you.
All right. So basically, you know, love it. Sounds great. You know, please specify computational challenge. Right. Okay, find novel regions of heart tissue multiomics using GWAS, scRNA-seq, rentiation. What's rentiation? Farhan, are you still there?
Yeah, I mean, that's not the text that I uploaded there, honestly.
Chemo treatment?
That's not my text.
Okay.
I don't know what's happened there. Sorry for that.
Okay. So there may be another deletion. Oops. Maybe it was a terrible idea.
I did.
Is it possible that it's readiness because the last word is like differ? It could be like differentiation.
Ah, thank you. What a good one. I love that. And Farhan, is that yours? Yeah, that's
Yes.
yours.
Yeah.
Nicely done, Mr. Detective or Miss Detective here. So Tessa, was that you?
No.
Who was saying that? Who was speaking?
I was.
Oh, that was you, Sonia?
Yeah.
Nicely done. The beauty of having a lot of eyes looking at the same problem.
Yeah, thank you.
All right. So yeah, that may. There you go. Good. Yeah. So what I would say is somehow combine them. I mean, they're all great, right? So
Yeah.
They're all in the same theme. So
Mhm.
All right. So Sonia, you've earned your way to go next. Let's see. I'm searching for your name. There you go. Okay. The paper I read last week, it developed. So let's see, Sonia. Yep. They developed a workflow called CNIC that can infer gene regulatory networks and infer cell states using single-cell RNA-seq. Takes in the gene expression matrix after a scored matrix, and then identify cell types hierarchical version. Test out everything hybrid methods to improve the outcome. Sure, sounds great, but where is the computational challenge? How can you benchmark it? This would benefit greatly from a complementary dataset. For example, I don't know, suppose that Farhan had a bunch of stuff in heart, and you now said, okay, great, let's now combine this single cell regulatory network approach with the, sort of hard data sets, then you can validate and so on and so forth. So basically, could combine with a classmate that has an awesome sort of biological problem in search for cool integration methods. And then sort of that will provide a natural place for expanding would provide natural place for expanding the Anyway, so there's like a gazillion typos here. Okay, does that make sense? So,
Yeah.
predict the best, most suitable treatment, medicine, pain medication for a type of cancer based on single-cell RNA-seq data on tumor cells. Extremely challenging. Need to know target genes for the pain medication in the first place. We could still look for overlap, but how could you validate that what you're real. So basically, so an alternative is if you know pain medications that work well with specific cancers, and if you know their target genes, could build a predictive model that says, could I have predicted these drugs would work best using the gene target overlap. Does that make sense?
Yeah.
Apply scenic to neurodegenerative diseases, Parkinson's, to infer gene regulatory networks and cell types affected and compare results previous studies by novel gene pathway enrichment. Using those results from scenic EHR, multimodal predictive model. Yeah, I love it. So here I would put Alzheimer's and Huntington's disease. And the reason is that we have scRNA-seq data for both AD and HD. So you can see if you could have predicted the changed genes in the state at single cell resolution based on I'm going to try something different. Let's see. Tools, voice typing. The differential expression of these genes in bulk RNA sequencing data, comma, see if the deconvolution of these data and their projection into single cell space using the scenic algorithm allows you to infer the single cell data driven expression patterns. All right, I don't know how horrible this is, but anyway, do you get what I'm getting at, Sonia?
Yeah.
Yeah, so basically, if you focus on AD and Parkinson's, come on, and then you can sort of actually validate using the single cell data. Sounds good?
Good. Thank you so much.
Of course. Lily, actually, let's do Landon. Landon, are you there?
Yeah.
Okay, awesome. Systematically rectifying errors in SARS-CoV-2 metadata. Pandemic has prompted unprecedented rapid sequencing effort. Genomics, often underappreciated. Metadata is exceptionally valuable, powerful tool for journalists. In spite of this, written with misspellings and inconsistencies. Submitting lab categories, multi-modulated approach semantically correct these, clustering isolates several pieces of metadata and identifying possibly originated lab and submitting lab naming consistency with the clusters essays for responding. So I like the idea of using genomic data to drive clustering that will then be used to correct phenotypic data and metadata mislabelings. However, I am concerned that this is basically an exercise in data curation and lacks a deep computational basis behind this that can build on that, period. Yes, this could be the hard part of the project that you're proposing, and you just have to do a lot of data curation before the fact, comma, but I'm concerned that your entire project will be the data curation part and will not be as satisfactory. I wonder who mislabelings is.
The mislabelings. It's like, it's crazy. People are just like are screwing up the data a lot.
Yeah, but you see my concern, right?
Yeah, no, I see what you, yeah, I mean, that is most of the project.
Yeah. So anyway, not thrilled basically. But anyway, it's not, I mean, if the clustering is really driven by genomics, then it might be kind of cool. But what I'm concerned about is that the true biology will be lacking. Unconventional signature of convergent evolution across mammals, independent evolution. However, overwhelming majority of studies interrogating convergent evolution, focus on scanning for convergence in coding regions. Dysregulatory elements. I like it a lot, but again, it's very open-ended and hard to validate. Again, not everybody has to have a primary project. I think it would be great for some of you guys to join another project. So, yeah, that would be my reaction there. You too. Yeah, so my concern there is hugely challenging. Any folks before you have grown old and tired to try. I'm exaggerating a bit, but you see my concern. So what I would say is that all three ideas are awesome, but super, super challenging. So I would say, you know, look for an awesome partner, and, you know, it's okay. Are you comfortable with that, Landon?
Yeah, that's fine.
Okay, sounds great. So for those of you who want to go next, please turn on your videos. For those of you who have gone already, please turn off your videos, and then folks with videos will show up. I see Na and Lily, but I know them well, so I'm trying to go with people I don't know first. I'm sorry, guys. I'm being unfair to you. So I see Mildred and Alice, and Manu and Joe. So Lily and I, is it okay if you guys go later? Do you have to go now soon?
No, that's fine.
Yeah,
okay, perfect. Thank you. All right, let's go with Alice first, and then Mildred, and then Manu, and then Joe. So Alice. Perfect. All right, the impact of, oops, sorry. Oh, I love this paper. This is great. Okay. Conducted their analysis solely on the individual European ancestry, making this profile more diverse to include other ancestry into the analysis. We will compare differences. However, the data collecting step might be challenging. Yes, I completely agree. Since we made so I love the paper. Of course, you can't collect more data. It will take a consortium. But you can build on the collection and integrate with more data sets, or be analyzed with full state-of-the-art deep learning method. Oh. Okay, does that make sense, Alice?
Yes, thank you very much.
Yep. I mean, again, I think it's a great topic, great paper, but, you know, I would look for something along these lines. Using the single-cell transcriptional data to understand cell states and therapy-induced evolution of the biological heterogeneity of diseases. From this paper, we can further uncover this process of advanced states. Weaknesses might be hard for cancer patients to get a biopsy during the treatment. You know, may become a useful way to track the evolution of the disease. But the problem is that the single-cell transcriptional data is actually coming from the oh, come on. Is that the one? No.
No, that's fine.
I'm sorry, the link is embedded in Harvard. I don't even know the
Sorry about that.
Oh, that's the one, right?
Yes, this is the one.
Oh no. No. When I do Control A, it selects the whole paper. Sure. Okay, evolution human long single cell RNA sequencing. Biopsies 30 patients before and during targeted therapy. So basically, these are usually operations that are done in the patient. So basically, you take the biopsy. Yeah. So again, great topic. Evolution of biological heterogeneity disease. Yeah. So basically, I think what you're suggesting is that somehow single-cell transcriptional data can be obtained without a biopsy, but it's only obtainable through a biopsy.
Okay, I was thinking like since that for patients with certain cancer type, they might be hard for them to like gain a biopsy like during their treatment over time. Therefore, we might be able to use transcriptional data that were previously labeled with at a different stage of a therapy to use those as a way to try to understand the evaluation of a certain disease.
Yeah. Talk with Jackie. She has SCRNA-seq data during tumor immunotherapy progression. So you could chat with her.
Sure.
Anyway. We can use a computational tool, RegNet driver, to identify regulatory cancer drivers. Yeah, Ectacorana. Yep. Thyroid cancer. So talk to Jackie, and then she can guide you with various stuff.
Sure.
Okay.
Okay. Thank you very much.
So we said then Mindran. So Mindran. Okay. In light of the ongoing pandemic, the SARS-CoV-2 genome browser and other sources holds algorithm sequence alignment study its evolution. We've done at the genome level. Sequence treats proteins at the protein level, especially examining those labeled mystery proteins whose function may still elude us. So we've been classifying mutations based on their evolutionary history. Yeah. I think after you've read this paper, you'll have a lot more insights and ideas. Does that sound good, Major?
Yeah, thank you.
AlphaFold, deep learning for protein structure predictions, entirely yeah, so you could combine with the previous paper to look at prioritization of mutations based on their folds, potentially even combine with Young Rice paper to see if mutations that perturb the predicted protein structure have been excluded from sarbecovirus. Does that make sense?
Sorry, could you clarify that?
Yeah. So basically what I mean is that suppose that you have this cool tool for predicting protein structure with AlphaFold. So maybe you could get annotations on the primary genome sequence for where are particular types of folds. And then you can go across evolution and then look at what are the regions that are evolving faster or slower, and then see if those regions somehow coincide with the ones that are within particular folds. Does that make sense?
That makes sense, thank you.
Okay, great. So basically what you could do is run AlphaFold. Sorry. Okay, is that okay?
Yeah, that sounds great, thank you.
Okay, great. Number 3, modern medicine, great emphasis on extending lifespan, and health span. This paper discusses available data sets, approach for computational tackle the problem of aging, and this discuss hallmarks of aging to the genomic stability. Yeah, so sure, you could talk to Elvira Kinzina group. Aging has predictions of deconvolved expression from GPEX data. This is one possibility. Okay.
Okay, so just contacting them about like obtaining this sort of data and then.
Yeah, and just sort of bounce ideas with them.
Okay.
Yeah.
Great. Yeah, thank you so much.
So who was next? Manu. All right, so idea 1, modeling statistics in methodological networks. Random matrix theory is a set of mathematical tools and analysis used to analyze systems in theoretical physics. I can find the matrix also in the interest of random, new papers, biological network for random matrix theories, with Python study. Biological networks, a couple of examples. I would like to build a set of tools that can be used to model study similar matrices in biology based on existing approaches. And study denoising, predict expression changes in healthy and cancerous tissues to improve quality sleep. Sure. Computation will be tricky, but seems challenging. Does that sound good?
Yep.
Latent space models incorporate both structural and sequence information proteins. Computational effect of proteins and protein stability. In theory, use this design to new stable variants to design new stable variants for protein. Energy-based approaches to protein design, Rosetta-based approaches. It's energy function from AI team so that you can learn energy function from multimers. Direct and close behind neural network-based energy function, expensive energy calculations, protein structure. Incorporating energy function, information latent space, anti-neural structure, collecting theory permutation complexes, and use them to train a neural network-based energy function like the paper, designing better therapeutic targets. Wow, seems extremely challenging. Yeah, there's just so many parts, and it's very, very challenging. So I would, caution you against it.
Okay.
You guys can always prove me wrong. So I just don't take no for an answer, basically. But it's just a cautionary. Protein DNA interaction networks, DNA binding proteins that we can collect from a bunch of papers. We can use this information to design a machine learning algorithm that uses the newer language-based models to predict DNA binding proteins. Using a combination of these approaches, we'll be interested to some of the effects of fermentation. Okay, so C work by C.
Yeah, Tosh is actually used to be a student in our lab, just kind of like improve,
Sorry, say it again?
Thrash used to be a student in our lab.
Oh, that's right.
Yeah.
So what do you mean by language-based models? Is that one of the citations here? Or is that?
So yeah, it's kind of like using existing like protein like the Facebook papers kind of an approach to specifically. I guess like here I'm not very clear on the architecture by itself. I was trying to approach two different topics. One was basically testing whether the putting like latent spaces that we already have from Facebook and also from the Church's lab to see if they can actually design mutants of protein DNA binding things. Like does the latent space actually find like mutational effects that are beneficial for binding with DNA? That was like the first question I wanted to answer here. And the second question I wanted to see is that can you incorporate information about the DNA sections that this thing binds to along with the protein sequence information to build a latent space that I can just sample around. Basically, if I have a protein that already binds with the DNA that I know, can I just like sample around it to find a mutation that binds much more tightly?
Yeah. So if you feel that you have the, make sure you have the right guidance, and then also relevance might be tricky since mostly proteomics hard to build on class topics. Right? So these would be my concerns. Okay. I'm sorry I can't be more positive, but again, we can't, we have to, it's like 1 idea out of 9. We can't select 9 out of 9. Okay, so who was next? So wave your hand if you were next. Okay, so Evelyn, you were next?
I'm not sure if anyone is before me.
I think I had mentioned 2 more names. So I think Joe was mentioned. And then Audrey, had I mentioned your name?
I don't think so, but I can wait.
Okay, sounds good. So let's do Joe, Evelyn, and then Audrey. Is that okay? All right. Drug repurposing, graph neural networks. For the paper summarizing new network inference and drug repurposing pipeline have been developed in very druggable models. Multimodal omics and drug screens. Uncover hub-like molecules of gene networks, the strength and challenge of this. I'm looking at the time. We only have 3 minutes left. Let's see. We only have, I guess, if we have quite a lot of people. Oh my God, I realized that it's dark in my room. So, I kind of have to go in 2 minutes. So I don't know how to do this. One way is to do it offline. Did you guys feel that it was helpful to do it online? So being able to chat is probably a good idea, right? So should we just do that again, I guess tomorrow or something like that? You guys want to do another mentoring session tomorrow?
I think for the students who sat through today's meeting, though, we should probably jot down their names in case other people show up tomorrow.
Yeah. So let me take a screenshot of the folks who are waiting now, and then we will start with you guys. So basically, we'll do 1, 2, since you guys were waiting the most, and then 3. Actually, we just did, Joe. Did we do? No, we didn't. 6, and then Ari, we've already done you, right? So, and then Kathleen, you've already gone, right? And then Sonia went. So Ari, Kathleen, Sonia, and then Christian, have you gone? And then Alice is gone.
Yeah, Professor, sorry.
So Christian, you haven't gone yet, right?
I have not gone yet, yeah.
Okay, so basically, oh, let me share my screen. This is what I wrote down. So basically, Lily, Na, Joe, Audrey, Evelyn, and then Christian will go next. Is that okay? Yeah. Awesome. Thank you, guys. Again, I hope that you're, so let me do a poll here. So basically, who feels that they're learning when I'm giving feedback to other people? So do you feel that you're learning when I'm giving feedback to other people? Okay, good. This is good. Awesome. So I guess you guys did not all waste your time. So the answer is 52200. And then and then the next question is, do you guys feel that this is a useful exercise to have a chance to go through your proposals with me directly? So let me, so not just about other people, but now about you. Do you feel that this exercise is helpful for you when I'm talking about your project? Okay, good, even more so. So, okay, so we'll do that again tomorrow. I mean, again, I'm sorry for giving you so much feedback so early in the term, but I think it's so helpful to sort of get that now rather than the last day of class when you're presenting your project. So I will put this for the next, oh gosh, there's a gazillion. Yeah, some things don't scale as nicely. I think peer review scales nicely, but this doesn't scale as nicely. But at the same time, I hope that this is helpful, that you guys. So the session will be at the same time tomorrow. So basically, I added it to the group calendar at 5:30 to 6:30. So right after Brian's office hours. Is that okay, guys? Yeah. Go ahead,
yeah, so I'm 4th, but I don't think they'll be able to make it until a half hour in.
Yeah, that's fine.
Okay.
You know, basically we'll look for you, and then when you show up, you just wave and say, "Hey, I'm next."
Okay, thank you.
Okay, awesome. Great. Well, thank you guys, and see you tomorrow. Bye bye.
Thank you.
Thank you. Bye.